It is a great pleasure for me to share my perspectives. I've been given, let me share my screen. I have been given 30 minutes to talk about targeted therapies and progress. So, of course, I cannot go into all the details, but I can give an overview and some perspectives from my end. I'm originally from Copenhagen in Denmark. I moved to America in 2000 and was in University of Colorado for 18 years, professor there. I was the CEO of the World Lung Cancer Organization, IASLC, which I actually developed already from my time in Copenhagen. And during my development of IASLC, I made close contact with the Chinese community. Over the last 5 years, I have been executive director for Mount Sinai in New York. And let's see here if I can move it forward. Why can't I move it forward? Here. I have some disclosures. I'm in advisory boards for many companies, and I have also a patent. So let me start with non-small cell advanced disease. And again, the focus today is targeted therapies and not immunotherapy. So you have seen this pie chart many times, and the pie chart is developing the whole time, almost week for week. You have new targets, you have new drugs. And we can see here we have many targets, we have many drugs. We don't only have first generation drugs, we have also second generation, and in some cases, which I will come back to, third generation and even fourth generation drug development within each target. Let me use EGFR as a story. We are this year celebrating 20 years discovery of the EGFR mutation, and we all know what EGFR mutation has brought to the field, particularly in China and in Asia. But osimertinib is the third generation EGFR TKI, and the PFS for osimertinib was 18.9 months versus first generation TKI 10 months in the FLORA study, where osimertinib was studied in monotherapy, a median survival 38.6 months versus 31.8 months. Then we are starting to combine osimertinib in FLORA2 with chemotherapy, and we can see the data there for the median survival for FLORA2 is not even reported yet. The osimertinib is around 36.7 months. And for Mariposa, which is a further development, is a bispecific antibody, amivantamab, with targeting both EGFR and MET, and we can see PFS benefit there compared to osimertinib alone. So we are starting from EGFR TKI monotherapy in several generations. Now we are starting to see combinations. And most recently, I just picked it up today in Journal of Thoracic Oncology, real-world data for osimertinib alone, and we can see 5-year survival is 18%. So how do we choose one option to the other? We have osimertinib alone, we have osimertinib in combination with chemotherapy, we have amivantamab and lazertinib combination. How do we choose for the patient? And that is a difficult thing because we have to weigh the toxicity and the efficacy here. The ideal scenario is, of course, high efficacy, low toxicity, low cost, and highly convenient. We cannot get everything. With osimertinib, we get moderate to high efficacy, relative low toxicity and low cost, and highly convenient. With osimertinib, with chemotherapy, we get a higher efficacy in FLAURA2, but we also get a higher toxicity and a higher cost and less convenient. And with amivantamab and lazertinib, we get a higher efficacy, but certainly also higher toxicity, higher cost, and less convenient. So those are the things we need to think of and discuss with the patient when we have the patient. What will be the best choice for the patient? Been talking about EGFR. When I started the EGFR story, it was distinguishing whether the patient had EGFR mutation or not. We soon learned that EGFR mutation was not one thing. It was deletion 19, and it was point mutation exon 21, L858. So there we had two. Later, we have learned about resistant mutation. We started to learn T7090M, and based on that, osimertinib was developed. But we have now also exon 20 mutations and insertions, and it gets more and more granular. Another thing we are starting to learn more and more and has a clinical relevance is co-mutations. It started with the P53 together with EGFR, and we learned that if you have a P53 mutation together with EGFR mutation, you have a poorer outcome. And today, we know that the co-mutations play a role. We know that for KRAS, which I will come back to, and also for immunotherapies. Acquired resistance to osimertinib is something we need to focus on, and that led to amivantamab, the bispecific antibody targeting both EGFR and MET, which is an acquired resistant pathway to osimertinib. And we need to learn more and more about those resistant pathways and resistant mechanisms. Transformation to small cell lung cancer. We know very little about it, how the biology behind it and the treatment of it. And that is something we need to learn more about. We need also to learn about drug-tolerant persister cells. How do we identify clones which are already from the beginning resistant? If a tumor is heterogeneous, and if the tumor has 90% sensitive clones and 10% resistant clones, those 10% resistant clones will emerge to be 100% over time. So we need to figure out how do we identify drug-tolerant persisters and how do we treat them. And that is some of my perspectives for the future. So we're starting to learn more about clinical trials where we take the resistant pathway into account. We learned at last ASCO a few weeks ago, savolitinib, which is a MET targeted drug in combination with osimertinib versus chemotherapy in EGFR mutant and MET amplified non-small cell lung cancer, the SASHI study. And we saw very good response rate for Savo-Osi, and we saw also very good progression-free survival, even in patients with history of brain metastasis. So it will be interesting to see how that move forward in the future. And we are starting to see more and more clinical trials where we combine and target the resistant pattern to EGFR TKIs. One thing we are starting to learn more about is co-expression of targets for ADCs, antibody-drug conjugates. And there was a very interesting study a few weeks ago in Clinical Cancer Research where they have used fluorescence multiplexed assay to study the overlap of EGFR, HER2, HER3, TROP2, all are candidates for ADCs. And of course, it is important for us to know how those overlap, because if we in the future should make a rational combination of drugs, we need to know what is the overlap of the targets. And we can see here from the same study that in the EGFR space, there are also HER2 expression, TROP2 expression, HER3 expression. So we're starting to learn much more about it. Antibody-drug conjugates are rapidly developing, and we know the first generation and next generation ADCs. ADC has three main components. It has the antibody, it has the linker, and it has a payload. And we need to figure out how can we optimize the effect of those ADCs, and to be honest with you, reduce the toxicity. The toxicity is still very significant for ADCs. So we have different developments in the ADC. We have bispecific ADCs, we have dual payload ADCs, we have immune stimulating payloads, and we have radionuclide ADCs. Everything is in development. And this is a bispecific antibody. I used to show this slide because I love to see the kids playing with Lego. Lego is a Danish product, and I am from Denmark, so I'm a little bit biased, but I love Lego. And I'm sure you know all the Lego bricks. And we want to build the very nice house to the right. But before we come there, it is a kind of a chaos with the Lego bricks. And with ADC, I think we are only there in the development. We have a long way to go. We are starting to learn about combination of targets. We have EGFR HER3 targets, and we have anti-HER3 ADCs, etc., etc. Here is a commentary I made 2 weeks ago, I think it was, in Clinical Cancer Research. And the title of the commentary was Antibody Drug Conjugates in Non-Small Cell Lung Cancer. Where is the target and where is the biomarker? Is the target the protein expression? Is it the copy number? Or is it a mutation? Many of the ADCs are developed based on mutation. But mutation, in my opinion, they are highly associated to TKIs, but not necessarily to antibodies. Antibodies should, in my opinion, be more associated to protein expression or copy number. We don't know exactly what is a target and what is the predictive biomarker. Some studies have a predictive biomarker, some studies have not a predictive biomarker and is done in all comers. Example, HER3, example, TROP2. So I really don't know the answer to this question. What about the other oncogene-addicted non-small cell? We have EGFR talked about, we have ALK, we have KRAS, HER2, BRAF, NTRK. I don't have time to go into everything, but KRAS has undergone a development. It was undruggable for decades. We tried. We could not target KRAS, which is the most frequent abnormality in non-small cell lung cancer, beyond EGFR in Asia. And the most frequent subtype is G12C, but we have many other subtypes as well. And we know for G12C, we have seen development of drugs, first sotorasib, then adagrasib, and now further other drugs. And we are seeing also combinations with KRAS and EGFR antibodies. I know in China you have studies going on that. So we will see what that will bring us. ALK is a wonderful story, and it has gone on for many years in different generations. First generation crizotinib, second generation alectinib, third generation brigatinib, lorlatinib, and so. And we have here the different generations. And we learned last year, and I'm proud my previous fellow, Dr. Solomon, presented at ASCO, the CROWN study, the 5-year survival, and 60% of the patients are still alive after 5 years. It's unbelievable. When I started in oncology, the outcome for those patients were 7-8 months. Today, 60% are living more than 5 years. This is amazing. And we have now 4th generation ALK inhibitors, which seems to be very active, 40% response, even in patients who have progressed on lorlatinib. So that is amazing, and there is a lot to look forward to also there. And we have also ALK vaccine in development, and it will be interesting to see where that will lead us. Here is what I'm thinking the future will be for ALK-driven lung cancer. As we can see here, we have resistant patterns. They are sensitive to one drug and resistant to another, and resistant to another drug and sensitive to another, and so on. So it will be possible to sequence ALK treatment based on mutations. We are not yet there. At least in our country, we don't use mutation guidance for sequencing of ALK, but we will be there very soon. Also here, we need to talk with the patient. Every drug they have very similar efficacy, but very different toxicity, and that needs to be discussed with the patient. So that is my perspectives in brief for advanced disease. For early stage disease, we know that even in the most early stages, we don't cure all the patients. So we need to improve the treatment. And as this presentation is focusing on targeted therapies and not immunotherapy, I will start with the EGFR story. And we know all the Audora study, adjuvant EGFR therapy for patients with non-resectable local regional disease. And we can see here today we have long-term survival data, and even if the curves are starting to narrow, I am a strong believer that there is a difference in the survival curves between osimertinib and placebo. The question we are asking ourselves today is how long time should the patients be treated with an EGFR TKI in the adjuvant setting? And the answer is we don't know. Hopefully, a liquid biopsy, MRD, minimal residual disease, can lead us to a decision, but we don't know yet for sure. We have also neoadjuvant osimertinib with a good outcome. Those studies are still ongoing, but I'm very hopeful that we can move the EGFR story to the neoadjuvant setting. ALINA study for the ALK positive patients you should be very much aware of. Many Chinese institutions participated. Yilong Wu was a principal investigator, and we know that alectinib was much better than chemotherapy in the adjuvant setting in all subtypes. And even in the neoadjuvant setting, we see good results for alectinib. And new drugs are coming in. The NeoCOAST study are studying new drugs, as we can see here, oleclumab, monalizumab, Dato-DXd, and many other new drugs are studied in the neoadjuvant setting, and I think we will see much more about that in the future. Open question for early stage non-small cell lung cancer. What is best, neoadjuvant versus adjuvant versus perioperative? Perioperative means neoadjuvant therapy, surgery, and adjuvant therapy. And we don't know, they have never been compared, neoadjuvant versus adjuvant versus perioperative. That is type of the studies we need. Which patients will need adjuvant therapy? If you have a complete pathologic response after neoadjuvant therapy, do you really need adjuvant therapy? That is a question we don't have an answer to. We are currently studying that in clinical trials. What is the role of minimal residual disease? Can that be used for escalation or de-escalation of treatment? We talked about duration of adjuvant therapy. What is a predictive biomarkers? I'm primarily a biomarker person, and I'm very much focused on how can we select patients to neoadjuvant therapy, to adjuvant therapy, etc., etc. And we need new drugs. Local regional disease, finally, I should probably not discuss Pacific because that is immunotherapy, that is not targeted therapy, but there are a lot of questions also related to Pacific study. Adjuvant durvalumab, as we know, improves outcome and is now standard of care. But do all people need durvalumab? How long time do they need durvalumab? And of course, we were all puzzled by learning about Pacific 2, where immunotherapy was given together with radiotherapy, chemotherapy, not as a maintenance. And that study was negative, as you all probably know. And what is the reason for that? In my opinion, the reason for that is that the immunotherapy was given together, concomitant with chemotherapy, radiotherapy, but we need to learn more about the relation between CRT and immunotherapy. And the LAURA in EGFR positive treatment in the EGFR positive space, we saw that gave a very good progression-free survival compared to placebo with osimertinib. But osimertinib is given endless here. Is that necessary? How long time should we be given osimertinib? That is a question we need to figure out. So we talked about the questions, other targets and EGFR. We are in a time where other targets are being studied, ALK, KRAS, etc., etc. Just a few words about small cell lung cancer and where we are. I think the most exciting thing for me at this ASCO was the Delphi study with tarlatamab versus chemotherapy or standard of care in second line, and we saw the final data there. The overall survival was clearly better with tarlatamab, DLL3 T-cell engaging antibody. And we probably know, all of you know the story about tarlatamab, but this study here, I think, is changing standard of care and make tarlatamab a standard of care for second line. So there is a lot more going on in the small cell space. I was given 30 minutes to giving my perspective, so I think I will stop here. What we need is we need to work smart. We certainly need large studies, but I would like to see more smaller scientific focus studies where we learn the science behind the combinations, behind the sequencing of the drugs, and hopefully we will see that in the future. So with that, thank you very much for your attention. And this is a view from my office in New York. We can see Central Park to the right. I go there every day, and I welcome everyone to come and visit me in New York. Thank you very much for your attention.